high-grade serous ovarian cancer.
HGSOC?
Yeah, HGSOC. It's one of those acronyms that immediately just changes the temperature in the room when a doctor says it to you
Absolutely.
because it's aggressive.
Yes.
It's notoriously difficult to catch early. And perhaps the most frustrating part, and I know we've talked about this broadly before, is that we are still really struggling to treat the specific patient in front of us.
Right, rather than just treating the average case.
Oh, exactly.
And that is the absolute crux of the problem here. I mean, we talk about precision medicine constantly in oncology.
It's the buzzword of the decade.
Oh, totally.
Yeah.
But for ovarian cancer, the actual tools we have to predict who is going to do well and who's going to relapse are, well, they're blunt instruments.
We're basically relying on clinical staging and just seeing how much tumor is left after surgery, right?
Right. And don't get me wrong, those are decent indicators.
Mm-hmm.
Like, if a surgeon can remove all visible disease, that's great. But they don't tell the whole story. You can have two patients with the exact same stage, the exact same surgical result, and one responds beautifully to chemotherapy while the other progresses rapidly.
Because the biology is different.
Exactly. The biology is different, but our standard clinical metrics treat them as if they're identical.
So today we're doing a deep dive into a study that suggests the answer to this whole stratification problem, figuring out who is actually high risk, has just been hiding in plain sight the whole time.
It really has.
Specifically inside the routine CT scans that hospitals are already taking.
Yeah, so we're looking at a paper titled AI-driven radiomic risk stratification for high-grade ovarian cancer. And the premise is just fascinating.
Mm-hmm.
It's this idea that standard medical images contain data, patterns, textures, spatial relationships that the human eye simply cannot see.
Right.
But which correlate directly with survival.
And this is the field of radiomics.
Radiomics, yes.
And I want to be clear for you listening right off the bat, we aren't just talking about a computer looking at a scan and saying, that looks bad.
Yeah.
We are talking about converting an image into a massive data set.
Correct. It's mining. We are mining the image for quantitative features.
But here is where I usually get kind of skeptical, because we see these AI papers all the time, right? Like, AI predicts X with 99% accuracy.
Oh, not at a time.
But then you dig in and it's a black box, or it only works on one specific MRI machine in some specific basement in Boston.
Right. The generalizability problem.
Exactly. But this study claims to be an end-to-end framework. So what makes this one different?
That end-to-end distinction is vital, because most radiomic studies, they kind of skip the hard part.
How so?
Well, they start with a tumor that a human has already carefully outlined. This study claims to automate the grunt work of finding the tumor, first the segmentation, and then use that automated map to predict survival better than our current gold standards.
Okay, let's break that down. Because if radiomics is so powerful, if we can basically turn images into crystal balls, why isn't this the standard of care in every hospital right now?
Right.
Why is my doctor still just looking at the scan with their eyes?
It really comes down to a boring, practical human bottleneck, which is time.
Time.
Yeah. To do radiomics, you need to isolate the tumor. You need to tell the computer, analyze the pixels inside this line and ignore the pixels outside this line.
Which is the segmentation part.
Right. And currently, that requires fine manual segmentation.
Which means what, exactly?
That means a highly trained radiologist sitting at a workstation going slice by slice through a contrast-enhanced CT scan.
Wow.
And remember, the CT scan is a 3D volume. We're talking about dozens, sometimes hundreds of slices for a single scan.
Right.
They have to manually trace the outline of the tumor with a mouse on every single slice.
Yeah, I can see the problem there. In a busy clinical workflow, nobody has 2 hours to sit and meticulously trace a tumor for just one single patient.
It's just not happening. It's expensive, it's tedious, and frankly, it's prone to variability.
Because humans are human.
Exactly.
Yeah.
If I trace it and you trace it, we might draw slightly different lines because tumor boundaries can be really fuzzy.
Sure.
So the authors of this study said, look, before we can even talk about predicting the future, we have to solve the labor problem. We need to automate the tracing.
And they did this using something called UNet-based architectures.
Yes.
Now, I've seen this term pop up in biomedical imaging literature a lot. Can we demystify that a bit? Like, what makes a UNet special compared to just a standard image recognition algorithm that, I don't know, recognizes a cat on the internet?
Right, the classic cat identifier. So without getting too bogged down in the math, Think of a U-net like a funnel that goes down and then comes back up.
Shaped like a U.
Shaped like a U, exactly. The first half of the network, which is the encoder, compresses the image to understand the context. It looks at the big picture, like, okay, this is a pelvis, this is an organ, this is bone.
So it's simplifying the data to understand where things are generally.
Exactly. But then the second half of the decoder expands it back out to the original size to localize precisely where the pixels of the tumor are.
Ah, okay.
It combines that high-level context with the fine details. It's designed specifically for this kind of masking task, where you need a pixel-perfect map of an object.
So it's not just classifying cancer, yes or no, it's actually painting a map.
Precisely. And to train this, they didn't just use a handful of scans. They used a massive data set.
How massive?
605 contrast-enhanced CT scans.
Wow, okay.
Collected over 14 years, from June 2004 all the way to July 2018.
And critically, and this is the part I always scrutinize the most with these AI papers, this wasn't just one hospital's data.
No, it wasn't.
Because if you train an AI on data from just one hospital, the AI just learns the noise of that specific scanner, right? Like, it learns that scanner A always has a slightly greenier image in the left corner.
100%. That's called overfitting to the source. It's a classic pitfall in this field.
Right.
If you train on Hammersmith hospital data and test on Hammersmith data, you get a great score. But the model completely fails the second you take it to a hospital in New York.
But they avoided that here.
They did. They used a multi-cohort setup. So they used Hammersmith Hospital in the UK for the development and training.
They split that one 7-3, right, for training and validation.
Exactly, a 70-30 split. That's where the AI basically went to school.
Okay.
But then they tested it on completely unseen data from two other major centers: the Cancer Imaging Archive, TCIA, in the USA, and the Kliniken Essen-Mitte, or KEM, in Germany.
So the AI is trained in London and then virtually flown to the US and Germany to take a test it hasn't studied for.
That is the ultimate litmus test for generalizability. If it works there, you know you have something real.
Okay, so let's grade the AI. It tries to draw the tumor using this UNet architecture. How do we actually know if it did a good job?
They used a metric called the DICE score.
Which, despite the name, is not a gambling reference.
No, though in research it sometimes feels like gambling.
Fair enough.
Think of the DICE score as a measure of physical overlap. Imagine I have a transparency sheet with the expert doctor's drawing of the tumor and another sheet with the AI's drawing.
Okay, I'm picturing it.
If I lay them on top of each other, a score of 1.0 means they are a perfect match. Every single pixel aligns.
And a 0.
A score of 0 means no overlap at all. They completely missed each other.
And what's considered good in the medical world? Because, biology is messy.
It is very messy. Ovarian cancer isn't a perfect sphere. It spreads. It's highly irregular. Generally speaking, anything above a 0.7 or 0.75 is considered pretty respectable for complex tumors.
So what did the UNet score?
In the Hammersmith validation groups of the home turf, it hit a DICE score of 0.90.
Wow, 0.90 is incredibly high. That's basically human-level variance, isn't it?
It is. It's essentially indistinguishable from an expert radiologist.
That's amazing.
But the real question, as we said, was the U.S. and German data.
Right, the unseen tests.
In the U.S. cohort, the TCIA, it scored 0.88. And in the German cohort, KEM, it scored 0.80.
Now, 0.80 is a drop from 0.90, obviously, but still well above that respectable threshold you mentioned.
Exactly. And that drop is entirely expected.
Because of the different machines.
Different scanner manufacturers, different contrast protocols, different patient populations. The fact that it stayed at or above 0.80 proves the model is robust.
It holds up.
It does. It effectively solves the bottleneck. We can now segment these tumors automatically, accurately, and instantly.
Okay, so step 1 is complete. The computer can reliably see the tumor.
Right.
Now we move to the core of this deep dive, the radiomics part. This is where we extract the data. But what are we actually extracting? Is it just the size of the tumor?
Oh, it's so much more than size. Once that segmentation is done, the algorithm mines the data inside that shape.
Okay.
They looked at geometric features, yes, but also first-order statistical features.
Meaning what?
Like, how bright or dark are the pixels on average? And then they look at the heavy hitters, which are the texture features.
This is where we get into things like heterogeneity, right? How chaotic the tissue looks.
Exactly. A benign cyst might look very smooth and uniform on a scan, like a water balloon.
Makes sense.
But a nasty, aggressive cancer might look chaotic, speckled, or rough, like a piece of gravel.
And the AI can see that?
The AI quantifies that roughness into exact mathematical values.
But I'm thinking, if you just unleash an algorithm on an image, you can get thousands of features.
Oh, easily.
And most of them are probably just noise.
That's a huge risk in this kind of research.
Yeah.
If you look for enough patterns, you'll eventually find a correlation that doesn't actually exist.
Like finding shapes in clouds.
Exactly like that. That's why feature reduction is so key here. They use 15 different feature reduction methods and 12 machine learning algorithms.
Wow.
They basically threw the kitchen sink at the data to filter out the noise and find the specific mathematical combination that best described the tumor's character.
They wanted to find a signature.
Exactly, a radiomics signature.
And the goal of this signature is to predict survival. So let's look at the actual results. Can this math predict if a patient will live or die better than the tools we use right now?
Well, the benchmarks here are tough. We have CA-125, which is the standard blood marker.
Right.
We have residual disease status. Did the surgeon get it all? And we have FIGO staging from the International Federation of Gynecology and Obstetrics.
Those are the current gold standards?
Yes.
And how did the AI do?
The radiomics model beat them.
It beat the standard of care.
It outperformed the standard clinical metrics significantly. It even outperformed a radiomics prognostic vector from a previous study.
That's huge. How do they measure that kind of victory mathematically?
They use something called the C-index.
We should probably break down the C-index for you listening, just so we're all on the same page.
Good idea. C-index stands for a concordance index. It's a measure of how well the model orders patients.
Okay.
Imagine you take two random patients from the study. One lived for 2 years, one lived for 5 years.
Okay.
Did the model correctly predict that the second patient would live longer? If it guesses correctly every single time, the score is 1 point well. A perfect crystal ball.
And if it's just guessing randomly?
If it's right, 50% of the time, the score is 0.5. A coin toss.
Okay, so random chance is 0.5. Where did the radiomics model land?
Radiomics alone scored between 0.60 and 0.72, depending on the cohort.
Which is pretty good.
It's very good. But the magic really happened when they combined the radiomics with the clinical data.
So they didn't throw out the doctor's notes.
No, not at all. They added the AI insights to them.
Synergy.
Exactly. When you combine the AI signature with age, stage, and residual disease, the C-index stabilized at 0.73.
Okay.
And crucially, it was 0.73 across all 3 cohorts: London, USA, and Germany.
See, that consistency is what really sells it for me. It's not a fluke.
No.
If it was 0.90 in London and 0.55 in Germany, I'd say throw it out.
Right. Back to the drawing board.
But 0.73 across the board suggests a real underlying biological signal.
It's highly robust. But I know a C-index of 0.73 sounds a bit abstract. Let's talk about what this actually means for a patient sitting in a consult room.
Right, the stratification part. High risk versus low risk.
So they used the model to split the patients into 2 groups using k-means clustering, high risk and low risk.
Okay.
And then they calculated the hazard ratio for those groups, adjusted for stage, age, performance, residual disease.
Now, this is the so what number. This is the number that makes you sit up straight.
It really is. In the Hammersmith cohort, the hazard ratio for the high-risk group was 4.81.
Nearly 5 times the risk.
Yes. If the AI flags you as high risk, you are nearly 5 times more likely to have a poor outcome, meaning progression or death, compared to someone with the exact same clinical stage who is flagged as low risk.
That is wild. And in the U.S. cohort?
It was even more dramatic, a hazard ratio of 6.34.
6 times.
Over 6 times the risk.
That is, I mean, that's not a margin of error. That is a completely different disease trajectory.
It really is.
Yeah.
Now, the German cohort was a bit lower, at 1.71, but still statistically significant.
Right.
But think about the clinical implication here. If I know a patient has a 6 times higher risk of death, I am not going to treat them with the standard watch and wait approach or the standard chemo protocol.
You're going to escalate.
I'm going to throw everything I have at them. I'm going to try to put them in a trial for a new immunotherapy.
And then conversely, if they are low risk, maybe you spare them some of the most toxic, aggressive treatments.
Exactly. That is the true individualization of care we talked about at the very beginning. It moves us away from this one-size-fits-all approach.
No, I have to play the skeptic again here.
Yeah.
Because this is the part of AI in medicine that always makes me nervous.
The black box problem.
Yes, the black box. We have this algorithm, it looks at a grayscale picture, and it spits out high risk. But does it actually know why? Or is it just picking up on something completely irrelevant? Like maybe high-risk patients tend to be thinner, so the AI is just measuring body fat. Or maybe it's detecting a watermark on the scanner software.
That is the question that kills most AI papers. Correlation versus causation. Is it looking at the biology or is it looking at artifacts?
Exactly.
But this study, they didn't stop at the code. They went back to the wet lab.
This is the part that frankly blew my mind. They actually connected the pixels to the biology.
They took the tumors that the AI flagged as high risk, and they did a deep biological workup.
What kind of workup?
Histology, transcriptomics, proteomics, copy number alterations.
Everything.
They wanted to know what is actually happening inside the cells of these high-risk tumors.
And they found a specific molecular signature.
They did. The high-risk tumors displayed a clear invasive phenotype.
Okay.
Specifically, they found dysregulation in SNARE interactions.
SNARE interactions. That sounds like a trap.
It's actually an acronym. Soluble N-acylmaleimide sensitive factor attachment protein receptor.
Wow. Let's just stick to SNARE.
Yeah, let's stick to SNARE. It's a complex of proteins involved in vesicular transport.
Break that down for us. What does that do?
Basically, it's the shipping logistics of the cell. It helps move materials from inside the cell to the cell membrane.
Okay.
In cancer, this mechanism is often hijacked to help the cell secrete enzymes that dissolve the tissue around it.
Which allows it to invade.
Exactly. It facilitates the physical spread of the tumor.
So wait, the AI saw a texture pattern on a CT scan, just a pattern of pixels, and that pattern turned out to correspond to a tumor that was physically better at invading its neighbors.
Yes, but it gets even more specific.
Really?
They also found activated MAPK pathways in those high-risk tumors.
Mitogen-activated protein kinase. I remember that from biology class. That's a classic cancer driver.
It is. It's the engine. It drives cell growth, proliferation, and survival. It's what makes the cancer aggressive and resistant to therapy.
This is incredibly important validation because it means the AI wasn't just guessing based on scanner noise.
Not at all.
It was detecting the macroscopic echo of a microscopic process.
That's a beautiful way to put it. The macroscopic echo. The chaotic division of cells driven by that MAPK pathway creates a specific texture, a roughness or heterogeneity that shows up on the CT scan in a way the UNet can measure.
Even if the human eye just sees a gray blob.
Exactly. The human eye misses it, but the math catches it.
So we really have closed the loop here. We have a system that automates the finding of the tumor, which solves the manual labor issue.
Right.
It predicts survival with high accuracy across 3 continents, solving the validation issue. And we know biologically why it works, solving the black box issue.
It's a very, very complete package. It addresses almost every major criticism usually leveled at radiomics.
So where do we go from here? What is the immediate application of this knowledge for you listening? Because I assume this isn't going to be in my local clinic tomorrow.
Not tomorrow, no. Regulatory approval for AI diagnostic tools takes time.
Sure.
But the immediate win is in clinical trials. Right now, ovarian cancer trials often fail because we mix high-risk and low-risk patients together.
Oh, I see.
If you test a new, aggressive drug on a mixed bag of people, the results just get washed out. The data is too noisy.
Right. But if we can use this AI to filter the patients first.
Exactly. You say, okay, this new drug targets the MAP pathway. Let's use the AI to scan everyone and only enroll the patients with that specific radiomic signature.
We could see much higher success rates in drug development.
Considerably higher. It allows for incredibly smart trial enrollment. And the kicker is, it's entirely non-invasive.
That is the best part. We don't need another biopsy.
No.
We don't need to cut the patient open again to find out if they have high MAPK activation.
We just need the scan that is already sitting on the hospital server.
It effectively turns a completely standard CT scan into a molecular diagnostic tool.
Which is transformative for global health.
Yeah.
It makes precision medicine scalable.
Because you don't need a fancy genetics lab in every rural hospital to run these complex proteomics tests.
Exactly. You just need a standard CT scanner, which almost everyone has, and an internet connection to run the model.
It really changes how you look at a medical image.
Doesn't it?
I used to think of them as just pictures, like a photograph you put in a frame, but they are really data sets.
Dense, three-dimensional data sets just full of biological secrets.
It leaves me with a thought, and I really want your take on this as we wrap up. We have millions, literally millions of CT scans, MRIs, and X-rays sitting in hospital archives around the world right now.
Gathering digital dust.
Exactly. Petabytes of data.
Oh, yeah.
If an algorithm today can look at a 15-year-old CT scan from 2004 and see the activation of the MAPK pathway, what else is hiding in there?
That is the billion-dollar question. We are likely sitting on a gold mine of diagnostic markers for heart disease, for Alzheimer's, for rare
Those
cancers.
waiting to be found.
We might find that the answers to some of our hardest medical questions have been sitting in the basement on backup tapes this whole time.
Waiting for the right pair of eyes, or I guess the right code to see them.
Exactly. The data is there. We just finally have the tools to read it.
Well, thanks for taking this deep dive with us. It's heavy stuff, but incredibly hopeful.
My pleasure. Thanks for having me.
And to you listening, keep asking questions. We'll see you in the next one.